I'm John Overington, and today I'm going to give a presentation about drug repurposing. I work at a company called Stratified Medical, but before working at Stratified Medical, I used to work at the EBI, building the Campbell database. So a lot of drug repurposing is trying to find and navigate this triumvirate between drug, target, and disease. And it's interesting to reflect on the number of potential objects in this sort of triangle. For the target space, there's currently about 400 human drug targets. So these are the targets through which current pharmaceuticals act. And these are selected from about 19 to 20,000 genes in the human genome. So about 1–2% of the human genome is currently a drug target. The number of diseases it's very difficult to estimate because diseases are really a spectrum and very difficult to draw boundaries around, but the numbers we use are about 5,000 for treated diseases, and there's about 14,500 diseases in total, of which about 7,000 of these are rare, so they occur in a very small number of patients, and traditionally have been neglected by pharmaceutical and biotechnology companies, but that's certainly changing. And then at the top of this figure is the number of drugs. So there's about 1,500 launched drugs. So these are chemically unique compounds or biological reagents used to treat disease. And these are sort of selected from or from a possible space of about 10 to the 20, 10 to the 21 drug-like molecules. So the chemical space, or the size of chemical space that we're potentially interested in mining is many, many orders of magnitude larger than the disease and the target space. So a lot of current research is really trying to, either given a disease, try and find some drugs and targets that link these two worlds up, or given a target, find a drug and disease, and then try and reposition that drug in a new disease setting. So a lot of the work that I'll tell you today was summarized in a publication last year, an open access publication, so you should be able to find this online fairly easily, written by an old collaborator of mine, Tudor Apriya, from the University of New Mexico School of Medicine, and myself. And in this paper we review various computational and practical aspects of drug repositioning. And one of the messages we wanted to put in this paper was that although you see a lot of publications around finding new uses of drugs or potential uses of drugs in a preclinical setting, the actual practicalities of making this therapeutically useful are incredibly difficult, but can be supported by informatics analysis, databases, and so forth. So in this paper we introduced this score, DREL, Drug Repurposing Evidence Level, ranging from essentially no evidence, just essentially in silico predictions with no confirmation, all the way through to level 4, which is actual experimental observation, a clinical observation, showing that the prediction or the drug is useful in a particular setting. So everyone's probably heard the story of sildenafil, Viagra, which was originally developed as an antihypertensive drug, and of course was later repositioned or reused and applied in the treatment of erectile dysfunction. So I won't bore you with that example. There's plenty of the story online. I'll highlight another couple of examples of successful drug repositioning in a few slides. So the nomenclature of the field is a little bit confusing as well, but I really use two terms, drug repurposing and then drug rescue. So drug repurposing is where you have an approved drug and you try and find a new clinical use for that. So because it's repurposing and the drug is approved, we know it's safe at the doses, and getting hold of substance to actually do either in vitro, animal in vivo, or potentially human clinical experiments is usually quite straightforward. Drug rescue is where you try and find a successful clinical use for a stalled compound, so something either currently in development that hasn't been approved, or was tried in a particular indication, a particular therapeutic setting, and failed in that use, but could be useful in a new application or a new patient subgroup. And the most interesting ones there are those that have got to fairly late stage trials, so phase 2 and beyond. And for these, we know that there's established pharmacokinetics, the drug's tolerable, so patients can take it without any short-term acute side effects, and the safety for this potential drug or this drug rescue has been established. And of course, there's little point discovering a drug or positioning a drug for drug repositioning if the safety profile is inconsistent with that actually known for the drug. And many reports, as we'll see, a couple of examples actually sort of showcase that. So drug repurposing is shown in this this diagram at a sort of high conceptual level. You have a virtual, so a computer-based store of drug structures, or an actual physical library, so samples in a series of test tubes or plates. And you have two things you can do with this. You can either run a virtual screen, so a set of target structures you get from PDB or model, and then doc structures in. You can apply fingerprint searching as well, and that's proved to be very successful as well. Then from the virtual screen, of course, you need to confirm the activity of these hits with actual experiments. So does the drug bind to the target? Does it modulate its function in the required way, and so forth? And is the selectivity that required as well for clinical use? Or you can go straight from the physical library and put them into a biomolecular screen, an actual experimental screen straight away. And from this screening, either virtual or experimental, you end up with a new target activity. You then want to confirm these clinical effects in a suitable model system or early human clinical trials, progress through to a phase 2B, phase 3 trial, and then really at that stage you have a repurposed drug. So you would have thought that getting the structures, the confirmed two-dimensional structures of drugs is a straightforward task. It's actually surprisingly difficult. A fair number of the structures in Wikipedia even are ambiguous or you know, in detail incorrect. And it's really an ongoing task for the community to work out and define the structures of these drugs in some detail. So here's an example of a real-world drug repositioning, and I've used the title here, "Some will pay for what others pay to avoid." And really here, this is the concept of sort of exploiting a desirable or undesirable side effect. So the drug we'll talk about is bematoprost. If you're familiar with the structures of natural products, the chemical structures of natural products, you can probably see that this looks a little bit like a prostaglandin. So it's structurally related to a prostaglandin. The drug name itself has got a prost section in there, so it's the name is, I guess, informing you that it's prostaglandin related. But this is actually a synthetic drug. So bematoprost was originally launched as a drug to treat ocular hypertension or glaucoma. And one of the side effects of this treatment for glaucoma, so high intraocular pressure, was the growth of fairly thick and vigorous eyelashes. And also the surrounding of the eye, the sort of skin on the inside of the eyelid took on a dark color as well. And if on the left-hand side of this slide I'm showing you the prescribing information, so that's the detailed description of the drug, the approved drug label for the drug, for Lumigan, which contains this bematoprost. And you can see here this warnings and precautions section, point 5 is pigmentation, and then for 5.1, and then 5.2 is eyelash changes. So this is a drug to treat ocular hypertension, glaucoma, and some of the side effects and the warnings to do with the use of this drug are to do with changes in the pigmentation of the eye and eyelash changes. Of course, people quite liked this side effect, and the drug actually was used quite widely off-label to improve the physical appearance of eyes. And so trials were then done. A new trade name, so trials for bimatoprost was done in this particular setting, and the drug was approved as a drug called Latisse. And if we look at the corresponding warnings and precaution settings there, you can see one of the warnings is an effect of intraocular pressure. So in one setting of the product, a side effect is potentially an adverse thing, and in another use of the drug, it's quite attractive. So a lot of drugs have really been like this. You can imagine that one of the side effects of sildenafil would have been unwanted erections. So hopefully it's an interesting example, a memorable example of drug repositioning. So a drug is launched, empirically some side effects are seen, and then these are then investigated, confirmed, show to be genuine, and a new use is found for the drug. But it's identical dosage and identical chemical structure for the drug in these two products. They've just got different names. So this is some of the marketing material for Latisse. You can see the clear effect on eyelashes in this particular photograph. So quite a striking clinical effect. When drugs are approved and when drugs are marketed, there's a lot of ongoing study of the drug to identify adverse reactions or side effects of the drug. So if a drug raises blood pressure, leading to a larger number of cardiovascular deaths, or causes heart murmurs or similar sort of adverse things, these are obligated to be reported to the regulator, and action is typically taken. And you can find pretty good side effect, adverse side effect data online. It's actually very difficult to find, primarily because it's not reported and there's no motivation to find positive side effects of drugs. So imagine a drug was launched, and then everyone on that drug suddenly lost about 5 or 6% of their body weight, but otherwise remained healthy. That typically isn't reported. I don't think it's ever been observed, but there's no real system or database where these sort of positive side effects are actually reported. It would be great if there was, but the sad truth is that they're not. So one of the challenges really of doing drug positioning is working out exactly what drugs are already used for, and this is primarily because there's no consistent description of disease and drug linkage. There's several classification schemes that exist, but it's really in the physicians, the regulators, and manufacturers' interest to stay silent on, the sort of a discrete and complete, comprehensive set of diseases. However, the drug label, those prescribing information documents that I showed you in the previous slide for Latisse and Lumigram, are the best guide for the approved allowed use. We found one of the best useful practical schemes to use is this World Health Organization, WHO ATC, so anatomic, therapeutic, and chemical classification scheme. And as I've said, the side effects in these drug labels are usually negative ones, and manufacturers need to be very careful about being perceived to encourage off-label promotion of their drugs. So WHO ATC is a comprehensive description or hierarchy of drug action. It contains combinations and also some odd things that we don't typically think of as drugs. And the other word or acronym that you'll often hear applied with the ATC abbreviation is the DDD, which is the defined daily dose. So the WHO maintain both a sort of hierarchical description of approved drug action and also store dosage, typical daily dosage information. And again, this is often quite useful in drug repositioning to work out, I guess, just how practical a new indication of a drug actually is. So the first level of the ATC looks a lot like the sort of departments on a when you visit a hospital: alimentary tract and metabolism, blood and blood-forming organs, cardiovascular system, dermatologicals, genitourinary, urinary system, and so forth. So this slide is taken from the Wikipedia page for the ATC classification. So there's a lot more background information there, but it effectively divides the body up. Or divides diseases up into a series of high-level top classes. The second level indicates the therapeutic main group and consists of 2 digits. An example is C, so cardiovascular, 03, and the 03 corresponds to diuretics. The third level of the code indicates the therapeutic or the pharmacological subgroup, and is made up of 1 letter. So C03C is a high ceiling diuretic. The 4th level is the chemical, therapeutic, or pharmacological subgroup, and is made up of a single letter. An example would be sulfonamides used for the treatment of used as diuretics, and these have the code C03CA. And then finally, the tip level, the leaf level, is the actual distinct chemical drug, and this consists of a further 2 digits. So C03, CA01 is furosemide, which is a sulfonamide, which is a high ceiling diuretic, which is a diuretic used in the treatment of diseases or some diseases of the cardiovascular system. And as new drugs are approved, they're added to these new nodes. And typically, if a drug is used in 2 or 3 therapeutic settings, it will have multiple codes. So this is the website for the WHO ATC and DDD database. You can search for drug names, you can use wildcards and so forth to pull back drugs. One of the other interesting things that people often are interested in drug repositioning is trying to find a use for a drug that's approved in one country and not another. Because the WHO covers the global health sort of environment, it can be a useful place to start to find drugs that are approved in say Japan and not approved in the US. And this sometimes can make the commercial application or the clinical application more exciting. So one of the key things that needs to be done in this processing or screening of this 1,500 set of drugs is to restrict the properties of that set so it's consistent with the disease. So probably most people would want to take an oral drug, something you can swallow in a tablet, something with no obvious safety warnings. Also required or wanted often would be low interaction with other drugs. So this DDI acronym, it stands for drug-drug interactions, where dosing one drug can lead to changes in the pharmacokinetics, primarily of other drugs. People probably don't want irreversible drugs. So these are drugs that bind covalently to their targets. Often these are associated with lower safety. Or are primarily applicable in very serious diseases for which there's no alternatives, like cancer. No controlled substances, So there's little point trying to reposition morphine or anabolic steroids without a lot of very careful consideration. Cytotoxic agents as well. So these are effectively drugs that kill cells, often selectively. But you know, you could probably reposition them in a new cancer, but it would be very difficult to put them in a sort of a chronic disease setting like hay fever, if for a drug that acts through killing cells. So what we use is ourselves is a filtered set of candidate drugs. These are drugs that have either got a USAN or an INN, and we annotate these with a few in silico models. Things like hERG is a predictor of, well, hERG is an ion channel in the heart that a surprising number of drugs modulate and can give rise to some quite serious cardiovascular events and deaths. The other model we use is blood-brain barrier model, this BBB acronym, and you either do want or don't want your drug to enter the brain, again depending on the sort of disease that you're after. And when we apply our filters on this set of about 12,000 compounds that have got a USAN or an INN, we end up with about 3,000 high priority or interesting compounds. One of the other features of drug repositioning, which is surprisingly common, is the fact that a lot of drugs, about 10–15% of drugs, are dosed in one form, one chemical structure, but actually are active in a different chemical form. And a nice example of this is the well-known drug tamoxifen, which undergoes two activation events shown by the heavier arrows in this slide. So there's a cytochrome P450-3A4 sort of demethylation to produce this N-desmethyl tamoxifen, and then a second metabolic event, an oxidative metabolic event mediated by cytochrome P450-2D6, introducing this OH to form the compound indoxafen. And there's a secondary route that this can happen as well. So tamoxifen can go through the first phenolic oxidation and then the desmethylation. So the key point here is tamoxifen it has some pharmacological activity, it's well absorbed and has defined pharmacokinetics, but the active component is primarily this drug endoxifen, which isn't present in the tablet, wouldn't be present in the drug screening file that you could think about doing virtual screening with, and so forth. There's other structure complexities to consider as well. For example, tautomers, epimerization. So this is where drugs change their chirality over time through just passive or potentially enzyme catalyzed events. Covalent reactive species, where a reactive metabolite is formed. An active metabolite, that effectively this is another form of where a drug is metabolized, often by a P450 to a different form, and this has either an improved or different pharmacological function. Hydration, enumeration of stereoisomers and so forth. Some drugs are dosed as a racemic mixture. The left and the right hand forms often have different pharmacological activity and certainly would dock differently into receptors and so forth. So dealing with all of these complexities is quite difficult to do well, and often increases the number of structures you need to consider very drastically, up from this 1,500 approved drugs or 3,000 high interest clinical stage compounds. An example here is midazolam. So midazolam undergoes a ring opening in the stomach. So about 50% of midazolam occurs in the left-hand structure form at low pH. And then there's a metabolite, so 10% of the total dose converts to this hydroxy metabolite. And probably, to stand the best chance of both understanding the positive and the negative side effects of a drug, you'd want to consider all of these chemically distinct forms. The drug discovery or drug targets often fall in distinct families. Currently a very popular and successful family is the protein kinase family. So these are often involved in signal transduction and inflammatory and cell proliferative effects, so application in primarily inflammation and anti-cancer settings. So we've put together a set of protein kinase, a database of sort of protein kinase inhibitors, specifically with the aim of looking at drug repositioning. 34 of these are approved. It's interesting to note that 3 of these are in Japan only, and 1 of them is in China only. So this gives some rough sort of indication of this opportunity between US, European approved and other parts of the world approved numbers. There's 498 of these in total, and we know the 2D structures for about 70% of these. And one of the issues there is that there's many errors in the structure assignment in the literature. So confirming the structure, if you're going to do any docking or other types of in silico screening, is essential to improve the likely success. And this set is useful in say, drug repositioning experiments, development of patient-specific treatments on the basis of genomic profiling of their tumors, and so forth. The process of drug repurposing can either happen through the original intended target for the drug, we call these on-target drug repurposing studies. And this is really finding new uses for the drug acting through this originally known target. And you can find new potential uses of that target or disease processes that target's involved in through the literature, through functional genomic studies, through genetic experiments, and so forth. And the big positive feature of this is that it's any subsequent use is likely to be, or more likely to be, compatible with the original dosing of the drug. Off-target repurposing is where you find new uses of a drug acting through a novel or unanticipated target. It is a fact that when you take a drug, it actually binds to lots of proteins in the body in quite a sort of time-dependent way, as we'll see. And this potentially is very exciting for quite radically different sort of drug repositioning experiments, but the big problem is that the drug wasn't originally optimized against that target, and so you need to be incredibly careful of dosing. The simplest case of drug repositioning is to take a library of drugs and to screen them in a model assay, and we'll look at 2 papers that did just this, both of them working in the area of neglected diseases. So before we do that, though, just a tiny little detour into drug action, where this really explains this on and off target effect, or the challenges of on and off target effects. So you swallow a drug, and then it's a fairly rapid absorption phase, shown in the red line. And then there's a slow elimination phase shown by the falling concentration over time. So the vertical axis is the concentration of the drug in the plasma, and the time is along the bottom axis. I've just noticed there that the first 15 should actually be 5. So the peak concentration of this drug is observed 5 hours, roughly 5 hours after dosing. In order to modulate the target, there's a concentration above which the target is effectively turned on or turned off, and I use the term XC50 for this, and this is the concentration at which the drug is efficacious against its intended target. And here we've got a drug that miraculously turns off its target for roughly 24 hours, so come the next day when you swallow the next dose of the drug, you can modulate the target again. So the acute drug adverse reactions, so ADRs, adverse drug reactions, are primarily linked to this peak concentration of a drug. And the unfortunate feature is that because of the differences in humans, their size, between a small baby and a large adult, and their gender in some cases, and their fat content in the body, and increasingly importantly is their sort of genetic differences. This Cmax can vary enormously due to but the drug dose and a wide range of environmental and genetic factors. And so some examples of these acute adverse drug reactions are this QT prolongation or hERG-mediated effect that I told you about earlier. An example of a drug here is cisapride. Another example, less inconvenient, is the blue or blurred vision that some people get after taking sildenafil, and although this is clearly an inconvenience, it's not very serious. But these acute drug reaction are often caused by the concentration of the drug in the plasma reaching a level where it actually modulates an off-target, so a non-intended target, and the drug just in the course of its dosing cycle reaches a concentration where it blocks the function or turns on, in some cases, the function of another target. Of course, because it's around the peak concentration, it's got a far shorter duration. So these drug side effects typically are observed shortly after dosing and then fall away or disappear, and the intended effect of the drug still remains because you're still above the concentration for the efficacy of the drug. So drugs, we know now, have got really a sort of pharmacology spectrum. So they bind to a lot of targets in the body, and a lot of the off targets that drugs bind don't really matter. They don't really do anything adverse to the body and under the time that they're modulated, at least by the drug. But occasionally there is an off target interaction that's associated with an adverse drug reaction or a safety risk. And there's a lot of research now in understanding and mapping the identities of those targets, the target organs they're in, and so forth. But a key feature of this is that as you dose a drug at higher levels, you get more pharmacology. So you have a richer set of targets that you modulate, and this is both positive and negative. So you're more likely to get adverse drug reactions, but you're also more likely to see other effects of the drug as well. So some of the ways that you can find these effects, as I've already told you, through the literature, through transcriptomics or GWAS experiments, and so bearing in mind the drug dosage is very important. In particular, the drug dose safety data that's known. The off-target effects won't be optimized. So if your drug repositioning or your potential drug repositioning is acting through a different to intended target, it's likely to have a shorter exposure in the body, and really is unlikely to be optimal for this new disease setting, but could be a great starting point for an actual drug discovery program exploiting this effect. Again, another really interesting thing about the field of drug repositioning is looking or trying to find out more about how drugs work, the molecular mechanisms, the safety, dosing, and so forth. The primary source of this information is the prescribing information. It's a term used in the US, or in Europe, this summary of product characteristics, or SPC. So when you have some time, maybe pause the video, look at take a drug name, abiraterone, lemborexant, and then look in Wikipedia, look in the prescribing information or the SPC to get an idea of the structure of these documents and the type of information they contain. At the time that I put together the slides. Interestingly, the structure of abiraterone in Wikipedia was incorrect. I don't know if it's subsequently been fixed, but you should be able to see or confirm the structure between the abiraterone prescribing information and that on the Wikipedia page. This is what a typical sort of website for a drug looks like, and as you can see in the top right-hand corner of this here, there's a link to the prescribing information. These are sometimes updated, or quite often updated actually by the regulators as new side effects or new data emerges about a drug. And again, they're a really useful, interesting source to do things like text mining on and data mining for a whole bunch of purposes. This is what the prescribing information looks like. It's got a particular sort of formal structure, so you can reliably find things in particular settings. It gives you a sort of edit history for the document as well. And these are sort of the ultimate or very important documents describing the action of a drug. So I thought it'd be useful to look at a couple of papers and potentially do a sort of virtual practical for these papers. So the first paper we're going to look at is a fantastic paper from some researchers in the States where they took a drug library and they screened this in a good assay of plasmodium activity, so potentially looking for antimalarials. They then looked into the action of these drugs at a sort of molecular fingerprint or a functional genomics response fingerprint level, found their mechanisms, and thought about identifying new molecular targets for malaria treatment. So this is an example where you have an assay, an experimental assay, in this case killing the malaria parasite, and you take a library of drugs, physical drugs, and do experiments, and they refound in the paper that antimalarial drugs actually were antimalarials in their assay, so they had a good control, and they found 33 compounds, I think, that were active but were not previously known to be antimalarials. So one of the key things you've got to do when you think about doing drug repositioning practically is think about this thing called a product profile. And this is really the sort of pharmaceutical requirements of a drug. You need to consider the delivery route. So this could be oral, parenteral. So typically this is injected. This is an injected form. Topical or inhaled. You need to think about drug-drug interactions. So some patient groups are on long-standing existing common therapy, and you need to make sure that there's no interactions with the other drugs they take. A nice example there would be warfarin. So if you've got a disease in a patient population where warfarin is likely to be used, warfarin is a drug that shows quite a lot of drug-drug interactions, and you would need to be very careful or considerate in drugs that you wanted to co-dose with warfarin. You of course need the drug to be safe, cost-effective, and efficacious, so it needs to be really better than what currently exists. And for diseases of the developing world, or neglected diseases, you need to think incredibly carefully about cost. There's little point repositioning one of these $300,000, $400,000 a year monoclonal antibody therapies for a neglected disease in parts of the world that the healthcare systems couldn't support the purchase of the drug. Dosing frequency is incredibly important as well. So some drugs you dose 3 times a day, some drugs once a day, others once a week, others every month. Ideally, for a lot of neglected diseases, you'd want a single tablet that was a cure as a sort of almost like a unicorn-like object or goal. But clearly, the more frequent the dosing, the more complicated compliance is. So the patient actually following the instructions or being able to follow the dosing schedule, and likely efficacy and safety. Cost of goods is also important. This relates directly to this cost of a drug. Another big area is the risk of misuse or the application of controlled substances. So a lot of drugs, the import and export, the handling of them is regulated, and these can be some quite fairly innocuous drugs in some settings. So avoiding any controlled substances is probably a good thing to do. You need tolerable side effects. Again, depending on the disease, safety in pregnant, breastfeeding, pediatric populations is incredibly important. A drug that requires very careful dosing or dose adjustment in pediatric patients, the safety data may not exist for that sort of application. So again, that's another sort of thing to think about very carefully. And again, for another sort of feature of some drugs is that they require sort of quite specialist dosing, or you need to perform specific tests to see how the body is reacting to the drug, say liver function tests. And so anything that requires monitoring, is, which would be visible on prescribing information typically, again, probably is a bad thing to have. So a task, which I think would be pretty informative to sort of think through some of the practicalities of drug repositioning, is the following. So pause the video, so this slide stays on the screen, and then spend 5 minutes developing a prototypical product profile for a novel antimalarial. Consider the dosing, so the dosage route, the type of distribution that the drug would want, the safety, the cost of goods, and patient population. And then in the next 30 to 60 minutes, go through the table in the Yuan paper that details the hits, and for each of these, try and classify the dosage, the safety, and the approval status for the drug to see how it matches your product profile. And then finally, triage the drugs to select that list against your sort of product profile to come up with the most likely repurposing candidates. And I think what you'll find is there's a really very surprising sort of message in this analysis, and that is that there's really no practical opportunities for drug repositioning in malaria against the set of drugs screened in this paper. So although they find a lot of actives, and although a lot of those actives are drugs, they're actually not very practical or useful for drug repositioning. If, like me, your product profile was a safe drug, so it's a very high tolerability, an orally dosed drug, so you didn't need to worry about injections or strange inhalation devices and so forth. And you wanted the safety because a lot of the patient population will be children or women of childbearing age. And of course, you'd want the drug to be approved. You wouldn't want to have to go through really expensive trials proving the efficacy. Ideally, you'd want to take a drug that's currently approved. So you'd want oral dosing, safe, and approved. If you apply that to that set of hits from the Yuan paper, you end up with two drugs that are oral, safe-ish, and potentially considerable for repositioning. These are the amphetamine and the drug orlistat, which is used as an obesity treatment in the West. There's a couple of obvious, well, there's a very obvious thing with the potential use of an amphetamine derivative, although we didn't think about adding, or I didn't add, controlled substance to the list there. That the practicalities of using a controlled substance, even if amphetamine was active, is just not even worth considering. There's a more subtle issue with the other drug that survives this product profile filtering, that being orlistat. So orlistat is an orally dosed drug, but it effectively has zero systemic exposure, so it doesn't enter the body. Its target is a secreted lipase in the stomach. This lipase chews up fats, lipids, makes them available to the body, and so it's a simple, the drug acts through a simple blockade of this lipase activity, stopping the body absorbing fats. So there's a couple of side effects of this. You know, the first is that I guess you're taking away a valuable food source in a often malnourished patient population with malaria, so that's probably not a good thing to interfere with anyway. But also the more practical thing is that the malaria parasite lives in the blood, and so if the drug isn't absorbed, it stays in the stomach. The drug actually has no exposure against its target system, and this is sort of fairly typical of the sort of attrition or failure to actually translate into something useful that happens during drug repurposing. You may want to help with your triaging. You may want to set a set of sticky labels, and for each drug, indicate whether they're approved or not approved, toxic or not toxic, orally dosed or not. So on this little coding scheme, aspirin would have a little tablet, a leaf, and a pair of lips for oral dosing. Maybe not the best. Set of images or selection of images, but with this sort of simple coding, you then be able to score and triage your set of drugs from this table in Yuan fairly straightforwardly. Another example, quite complicated, this one, but again from the area of neglected diseases. So this group at UCSF and collaborating labs set up a screen for the pathogen that causes amoebic dysentery. So this is a huge killer of in particular children in the developing world. And there's no really good current drug treatments for this disease either. So it's a great opportunity for drug repositioning. They followed a very similar strategy. They screened a drug library against an assay. The assay itself, because the amoeba has a sort of really complicated sort of lifestyle or environment required for growth. Actually, the physical implementation of the assay was very challenging. They found a relatively small number of active compounds as well, and in particular they analyze a drug, or propose a drug called Oranafin for the use of amoebic dysentery. But it's a really interesting exercise to read the paper, to then look at the prescribing information for Oranafin, do a little bit of reading around the drug action as well, and you know, certainly my reading of the paper is that Oranafin has got relatively little chance of becoming a treatment for amoebic dysentery, in particular because of the pediatric GI tract compromised nature of the patient population. So that's it, a sort of overview of drug repositioning. Some sort of context to the type of analysis that can be done either prior to virtual or physical screening, selection of drugs, the sort of factors and features that are required in drugs. Some pointers for how to get information on drug action, drug safety, and side effects. And then a couple of examples where taken from the literature, two high-profile papers. They got a lot of coverage at the time, whereby I guess we apply relatively simple filters and knowledge to filtering these sets to come up with a set of practical drug repositioning candidates.